Where do you get these files? You either go to our friend Ensemble, you go here for the FASTA file, and you go for the primary assembly, so this one. And then you go to the corresponding GTF file and you download it. Another possibility for human and mouse, that is the GenCode website. When you follow the link, you end up on this website, and here you can choose between human and mouse, as you can see. So let's go for human. When you scroll down, you get the FASTA files. So you have many FASTA files. What you need is this one, the primary assembly. So not this one, that's the one with also the haplotypes, with the alternate contigs. You need the primary assembly for RNA-seq. So the corresponding GTF file is here. When you scroll up again, you can find it here. So here is the corresponding GTF file for the primary assembly. What is this GenCode? Well, it's a consortium, so a large project that is coupled to the ENCODE project, and that provides annotation. So annotation for large-scale next-gen sequencing experiments. What kind of annotation? Well, all annotation that is linked to transcription. So not only the introns and the exons, but also the more complex stuff like the long non-coding RNAs, the pseudogenes, the microRNAs, and also very important, the different splice variants, which is really important in human and mouse. You have seen that. So for 20,000 human genes, we go to more than 200,000 different splice variants. So what are they doing? They are adding these annotations to the human genome and to the mouse genome. Where do they get the annotations of all these things? Well, basically they get them from Ensemble. Ensemble has what is called an annotation pipeline. So it's a sequence of tools that all will search for evidence in the sequence for the presence of exons, introns, UTRs, pseudogenes, microRNAs, whatever. So the predictions done by Ensemble are used by GenCode, but they are checking them. So they are adding manual annotation, so high quality annotation, that is done by the Havana group. And this is a group of scientists who are really going through the literature, for instance, to find evidence for annotations with respect to transcription. So it's the Ensemble annotation, but it's curated. A part of it is checked because there's a lot of information there. So it's higher quality than Ensemble. So I never go to UCSC, I never go to NCBI when I do RNA-seq. Why? Well, there have been many publications, and I listed 2 of these publications here on the slide, but there are more than these 2. Many publications where they compared. So in this one, they compared Ensemble annotation, so location of exons, introns, and so on, UCSC and RefSeq, that's NCBI, yeah, for RNA-seq experiments. And they came to the conclusion that when you work with RNA-seq data, that the best choice here is Ensemble. What's the difference between the 3? They all have their own annotation pipelines. And Ensemble is the least stringent of the 3. Ensemble will include all predicted annotations. If everyone finds them, it's included. But if only they find or predict a certain annotation, they also include it. While UCSC is much more stringent. It will only include annotations that all 3 of them find, so that everyone agrees on. They thought initially that this would be a better choice because you're more sure if everyone finds it, it must be real. But this paper comes to the conclusion that many of the annotations that are predicted only by Ensemble are real. Yeah, they exist. So Ensemble is the most complete one. This paper compared GenCode to NCBI. Yeah, it's not here, GenCode. And they also came to the conclusion that GenCode is way better for using in RNA-seq experiments than the RefSeq annotation. Again, because it's much more complete, it contains more transcripts, so more splice variants, more exons than NCBI. So everyone in the RNA-seq world kind of agrees that you go for Ensembl or for GenCode and not for UCSC. There is an additional issue with UCSC, and it's on the level of the counting tools. After the mapping, you're going to do counting. You're going to count the reads that map to a certain transcript or to a certain gene. And some of the counting tools also have an issue with the UCSC data. Why is that? Well, because in the Ensembl data, there are gene IDs and there are transcript IDs. And each transcript, so each splice variant, has the gene ID of the original gene. So the different splice variants that belong to the same gene, they will have the same ID, which is great for these counters. That's what they want. While on the other hand, in UCSC, the different transcripts that belong to the same gene will have a different ID. What is the downside for that? Well, they will be seen as multi-mappers. So if a read maps to a part that is common to different splice variants, so the different splice variants of the same gene, they have parts that are specific, but they will also have parts that are common. So if a read maps to one of these common parts, it will be seen as a multi-mapper and it will be thrown out. And that's also an issue. So second reason why you go for Ensembl or GenCode.